So today we are going to discuss about the structure of DNA. So before going to study the structure of DNA, one must know about the full form of the DNA and some basic knowledge. Just like the full form of DNA is deoxyribonucleic acid. See, it is considered as the genetic material that helps in the transfer of information from one generation to the next generation. Just like this is the one cell, and when it divides, then information that is travel from one cell to another, consider it like it is your first generation, it is the second generation, then the information that is travel from one generation to the next generation is through the DNA in the form of DNA. Okay, now what is the location of the DNA? See, I find the name of the DNA very interesting because of the one reason, and the reason is that it gives the complete information about the DNA that one must know about. Like nucleic acid, if the given is there, that means the location is located in the nucleus. Acid means it describe about the nature of the DNA, that DNA is of acidic nature, and since it is acidic in nature, so it has the negative charge over it. So this is the question, one question that is coming over here, that which charge is located or present on the DNA, and that is my negative charge. This is the concept of your NCERT as well, and this is the mostly asked question. Okay, now DNA is the biomolecule. We all know it is a macromolecule basically, that is made up of the smaller units, and the name of the smaller unit is considered as the nucleotide. Many nucleotide, what is nucleotide? We are going to discuss it later on, no doubt. Many nucleotide when collectively joined together, then we consider it as the polynucleotide. So here two words I have seen now, that is polynucleotide and deoxyribo. Now let's find out where we are going to deal with these two names. Okay, but before that, let's study about the basic components or the three main components of the DNA that helps in the formation of the complete structure of DNA. Okay everyone, so here it is, three components. Seriously, I really like those components. And first is sugar, my personally very favorite. Second one is nitrogenous base, another favorite of course. And the third one is phosphate. Okay, so sugar, we have pentose sugar, that means 5 carbon sugar in our DNA. Nitrogenous base, as the name suggests, the base in which the nitrogen group is present, and it is of two types. First is purine and second one is pyrimidine. Fine. And phosphate is phosphate, PO4 2 negative. Clear? Yes. Now the term, suppose students, if sugar and nitrogenous base join together, what is the term we are going to give it? Come on, we call it as the nucleoside. कैसे याद रखें? How do we remember this? Look, N means nucleo, connect कर देते हैं, N means nucleo, and side we get from the sugar. When sugar and nitrogenous base, sugar and any nitrogenous base when combined together, then it form the nucleoside. And sugar plus nitrogenous base plus phosphate group, collectively called as nucleotide. Remember the word nucleotide? From these three units when collectively repeatedly join together in a number, countless number you can say that, then we form the polynucleotide, that is our basic DNA structure. And we as all know that polynucleotide or DNA is a double helical structure. That means it has the two strands in it, okay, having the opposite polarity. Don't worry, we definitely discuss about the polarity and the strands of the DNA. But first let us study about the sugar. Okay, see, we have the pentose sugar, that is 5 carbon sugar. We study about the glucose, that is 6 carbon sugar. We already study about the 5 carbon sugar. We already study about the 3 carbon sugar, and so on. Okay, so the 5 carbon sugar are basically two. First is deoxyribose sugar and second one is ribose sugar. Simple difference, it is of RNA sugar, it is of DNA sugar. Both are 5 members, 5 carbon ring, nothing much. A slight difference in the structure, otherwise the structure is same as well. So this is the ring that I have drawn. I know the structure is little bit distorted right now, but it's okay. We have 5 membered ring. Look students, this is my carbon number 1, this is my 2, carbon 3, carbon 4, and here it is carbon 5. The positions that I deal with are second, third and fifth. Look at the second position. H and H group are there. No O, no hydroxyl group is present, nothing. So this type of sugar we consider as the deoxyribose sugar. You know, D means negative, oxy means oxygen, ribose means, in the ribose sugar, if we remove oxygen at second position, then it is considered as deoxyribose. And say people, if I add on O at the second position, then it forms the ribose sugar. Okay, and because in our DNA deoxyribose sugar is present, no oxygen is there, this is one of the major reasons for the stability of the double helical structure. First reason is obviously it is double stranded structure. Second reason for the stability, presence of, or you can say absence of, oxygen at second carbon of the sugar. Let's find out rest. Now nitrogenous bases. See, I already told you that we have two type of nitrogenous bases. First is purine. They are carbon and nitrogen containing heterocyclic groups. Purines and pyrimidine. Purines are also of two types, adenine and guanine, and pyrimidines are of three types, that is the cytosine, thymine and uracil. Okay. Now DNA has the very unique quality to form the bond in itself. Nitrogenous bases form the bonds by themselves by following a particular pattern. Okay, and that pattern is like adenine always pair up with T, that means the thymine, and cytosine always pair up with the G, that means guanine. Adenine can pair up with the T in case of the DNA, but if we talk about the RNA, adenine always pair up with the U. Let's not discuss about RNA main. We are going to discuss about the DNA only. See, here it is adenine always pair up with T or vice versa. T is always pair up with the adenine and G always pair up with the cytosine. It forms the double bond. You can see that the bond that is present, that is the double bond, and the bond between GC is called the triple bond, and these bonds are called the hydrogen bonds. Okay, that also provide the stability of the to the DNA. Clear, bachcha? Yes. Now phosphate, obviously that provides the negative charge to my DNA or to our DNA, obviously. So the stability reason third is presence of hydrogen bond that helps in the formation of the double stranded structure. Now look, we have deal with the three type of bond in the DNA mainly. First is hydrogen bond. Yes, where do we find the hydrogen bond? Between two nitrogenous bases like adenine pair up with the thymine and cytosine pair up with the guanine, or vice versa. Second one is, it is glycosidic linkage. Glycosidic bond is mainly for the sugar. So the sugar that is located is pentose sugar, that is the deoxyribose sugar. When joined together with the nitrogenous base, then the linkage is said to be glycosidic linkage. And now the third one is phosphodiester linkage. Hydrogen bond between two nitrogenous bases, glycosidic bond between sugar and nitrogen. But what about phosphate? Obviously, sugar joined with phosphate group with the ester linkage. So one sugar with two phosphate and one phosphate with two sugar unit. When this formed, then we call it the diester linkage. So this form the backbone of my DNA. Okay, so let's start to draw the structure of the DNA. But before that, obviously that 3' I told you that in sugar we need 3 main carbon, that is second carbon, in which we see that the hydrogen is present only, not oxygen. And third and fifth position, why? Look, this is my deoxyribose sugar. I'm not highlighting too much. This is my third carbon and this one is my fifth carbon. What would happen? Phosphate link with this third carbon and also with another phosphate with this fifth carbon. Okay, so that's why I'm saying one sugar, two phosphate. One phosphate, two sugar. Just like this phosphate that is located at the third end of the first sugar with the second sugar unit at the fifth carbon, it joins with it. It forms a ester linkage, two ester linkage, first, second. That's why we call that diester linkage. Clear? Yes. So this forms the basic backbone. Now let's start with the structure of DNA. Let's draw a little bit structure. Double helical structure. Simple. Draw sugar, phosphate, fish sugar, then phosphate, then sugar, then phosphate, then sugar. Similarly, sugar, phosphate, sugar, phosphate, sugar, phosphate, again sugar. and so on. Here it is 3 prime, then 5. Here it is 5 prime, here it is 3. So the polarity, that 3 prime, 5 prime, what is this? These are the carbon of the sugar unit. Okay, these are the carbon of the sugar unit that are flaunted at the ends. Clear? So this nitrogenous base, this contains post-adenine, this contains post-guanine, this contains thymine, and this also contains guanine. So alternate pair, which one? Thymine, cytosine, adenine, cytosine. Bonds, double bond, then triple bond, then double bond, then again triple bond. So this forms the structure of the DNA. Clear? So now the distance between two bases, this is first base, this is second base. The distance between two bases is 0.34 nanometer and total pitch of the DNA is 3.4 nanometer. The distance between two consecutive base pairs are 0.34 nanometer, whereas the distance between the pitch of DNA is 3.4 nanometer, and each pitch in the human B type of DNA contain 10 base pairs. See, what are base pairs? Pair means join. Okay, one pair means two. Okay, so here are how many pairs are there? 1, 2, 3 and 4. How many pairs? How many base pairs? We are drawing 4 base pair right now. So that means 4 base pair means how many nucleotides? There is a difference. Nucleotide single units are there, but base pairs means nucleotides are in pairing form. Okay. So how many nucleotides? 8 nucleotides are there. Okay. So this is the basic structure of the DNA. bDNA we are having in our body, that is the human DNA mainly. The pitch of the DNA is 3.4 nanometer with the 10 base pairs, and the distance between two consecutive base pairs are 0.34 nanometer. With anti-parallel strand, two strands are located and these two strands are anti-parallel with each other. 3 prime, 5 prime, 5 prime and 3 prime. That also provides stability to my DNA. So I hope the concept is clear to you everyone. All the best.